Welcome to our video on Arabidopsis leaf sterilization and microbial load counting. Remove plants from their growth chamber or other growth environment. For each sample, we will measure its mass to get a count of microbes per unit fresh leaf mass. Zero Weibo before sampling leaves. Cut leaves from the plants, leaving enough petiole to handle without damaging the leaf surface. Pat leaves dry using a lab wipe. Mask the leaves using the zeroed Weibo and record the value. Gently place leaves in the mesh cylinder. We have numbered the slots to keep track of the samples. Repeat until all the samples have been collected or all cylinder slots are full. Stack the cylinders and twist to combine. Soak the leaves in 10% bleach, 0.6% sodium hypochlorite for 1 minute with light agitation. Briefly shake off excess liquid, then place in sterile water and agitate. Repeat in a new beaker of sterile water. This should wash away the remaining bleach. Place the cylinder stack on the Weibo and remove leaves from the slots onto a sterile towel, keeping the leaves matched by sample. Pat the leaves dry and place them into sterile 2 ml tubes containing grinding beads. Prior to homogenization, the tubes have 250 microliters of 10 millimolar magnesium chloride added. Next, the leaves need to be homogenized. Place tubes into blocks, then load into the QIAGEN Tissuelizer 2. Other bead beating homogenizers should also work. Run for 1 minute at 25 Hz. Rotate the blocks, then repeat for even homogenization. Centrifuge the tubes briefly to collect the ground tissue at the bottom of the tubes. Fill a pipette basin with sterile 10 millimolar magnesium chloride. Add 180 microliters of solution to enough wells of a 96 well plate for 2 dilutions of each suspension. Dilute the suspension by adding 20 microliters of suspension to each well. Pipette up and down to mix. Then discard tips. Repeat dilution with the same volume. Prep R2A plates by adding a few 3mm sterile glass beads for spreading. Transfer 100 microliters of each dilution onto a plate. Shake the plates to distribute the solution, then collect beads and set the plates out to dry. After drying, stack the plates and store in sleeves. Incubate until colonies are countable. Protocol and demonstration by Hannah McMillan, and production, filming, and narration by Julian Liber. Music credit to Ann Joan. Thank you for watching!